Hello students, this is your Prasanna ma'am. Today I am here with the previous year questions of APEAPCET from the chapter Cell, the Unit of Life. So if we look into the most recent question which is asked in 2024, the first question which states like, to analyze the wrong statement from the following. If we see peroxisomes participate in photorespiration, he is saying photorespiration is also called as glycolate cycle. Photorespiration is also called as glycolate cycle or C2 cycle. Okay, and this cycle involves three organelles. They are, first one is chloroplast, then peroxisome, and third one is mitochondria. Third one is what beta? Mitochondria. Okay. So it includes three organelles. What are they? Chloroplast, peroxisome and mitochondria. If so, this statement is correct. If you look into the second statement, lysosomes perform glycolate cycle, he is saying wrong. Lysosomes are nowhere included in C2 cycle or photorespiration. Third one, endomembrane system includes endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. True. Alluroplast store proteins, absolutely correct. Alluroplast comes under the non-colored plastids. What are they? Leucoplasts. These are non-colored plastids. What is their activity? They store food. What do? They store food. If they are storing carbohydrates, then it is called as amyloplast. If they are storing proteins, it is alluroplast. If they are storing lipids and fats, they are ileoplast. So yes, this statement is also correct. What is the wrong statement here? The wrong statement is option 2. Okay. Next question. Which of the following is not the function of cytoskeleton? Cytoskeleton is a network of proteinaceous filaments. These are network of proteinaceous filaments which includes microtubules, microfilaments and intermediate filaments. Okay, and these microtubules, intermediate filaments and microfilaments together perform few functions, where the most primary function of the cytoskeleton include intracellular transport, then it also gives mechanical support. They help in maintaining the cell shape. Okay, and intercellular transport is not mediated by cytoskeleton. Intracellular transport means transporting the molecules or particles within the intracellular targets or within the cytoplasm of a cell. Intercellular transport means transporting the molecules or particles outside the cell or from one cell to the other cell. That transport is not mediated by cytoskeleton. Most important point to be noted, because cytoskeleton is asked twice in APEAPCET. Okay, you will see in the coming years also. Next question. Yes, here if you see, he is asking us to identify a membranous cell organelle. We all know that the most noted a membranous cell organelle is ribosome, which is present both in prokaryotes and eukaryotes. And the next a membranous organelle is nucleolus. Where do you find this nucleolus? This nucleolus is seen in a nucleus. Nucleus has four parts which include nuclear envelope, nuclear pores, nucleoplasm and nucleolus. What are they? 4 parts. First is nuclear envelope, nuclear pores, third is nucleoplasm, fourth is nucleolus. Nucleolus are amembranous and they are involved in rRNA synthesis. Okay. Cool. Now you see option is A. If you look into B, lysosomes are single membrane bound organelles, peroxisomes are single membrane bound organelles. Here again in option D, lysosomes are single membrane bound organelles. So what is the correct option? Option A is your answer. Next question, if you see, assertion and reasoning. In assertion, he said that ions and organic material concentration is higher in cytoplasm than in vacuole. It is wrong. If you consider a plant cell, 90% of the plant's cell volume is occupied by what beta? It is occupied by vacuole. What does vacuole consist of? Vacuole consists of all the excretory products, and vacuole consists of sap and all the excretory products. This is fully acidic. This is what? Acidic, and it has low pH, and it is highly concentrated. When you look into the cytoplasm, the fluid which is present in the cytoplasm has low concentration. Cytoplasm has low concentration, and when compared to this pH, here pH will be high. Okay. In that case, the transport of material from cytoplasm to the vacuole, the excretory products or the cellular waste or sap is being transported from cytoplasm to vacuoles through the what beta? Through the tonoplast, which is active transport. Which is what transport? Active transport. As this is against the concentration gradient, from low concentration to high concentration, as it is against the concentration gradient, it is called as active transport, wherein ATP, cellular energy, is required for this transport. Now if you look into assertion, concentration is higher in cytoplasm than in vacuole, he is saying. So this is wrong. Concentration in vacuole is higher than the cytoplasm. Next question, tonoplast facilitates the transport of ions and other materials against the concentration gradient. That is why ATP is required, and we are calling it as what beta? Active transport. Yes, reason is true. Assertion is false and reason is true. So the option is D. Next question, if you take next question, which is asked in 2023, we need to correlate the terms given in this box. Okay. If you see endoplasmic reticulum, it is discovered by whom beta? Porter. This and endoplasmic reticulum is involved in secretion as well. What type of secretion? If it is RER, rough endoplasmic reticulum, it is involved in the secretion of proteins. If it is a smooth endoplasmic reticulum, it is involved in the synthesis of lipids and steroids. So yes, here A, R and W. If you see ribosomes are given by or discovered by George Palliday, and ribosomes are purely involved in protein synthesis. Next, cell membrane, the most or widely accepted model of cell membrane is proposed by Singer and Nicholson, and he gave what model ma? fluid mosaic model. So here it will be C, P and Y. So if you take and list 1 A R W, B Q X and C P Y, option A is your correct answer. Next question if you take, which is from 2023 only, again one match the following type of question is asked from this chapter, wherein white blood cells are what? Amoeboid. Okay, mesophyll cells are round and oval. Okay, red blood cells, these are round and biconcave. Tracheid is elongated. So here if you see, what is the correct option ma? 3, 4, 2 and 1. Option D is your right answer. Next question. Again about cytoskeleton only you see. In 2024 also question is appeared. In 2023 also same question appeared. You see, cytoskeleton, of course it is involved in mechanical transport, cell motility, that is cell movement, not the molecules out of the cell, not intercellular transport. It is involved in cell motility, that is cell movement. And intracellular transport, though, it is the primary function of the cytoskeleton. It is not involved in cytokinesis. How it is involved in cytokinesis? That is an indirect function ma. For example, in karyokinesis, of course, for the formation of spindle fibers or spindle apparatus, microtubules or tubulin proteins are required. And for the at the time of cytokinesis or animal cell division, these are required, like actin filaments and contractile ring formation is happening, but that is outside the cell, not inside the cell. So in the direct sense, it is not performing the activity of cytokinesis. So when we consider these 4 options, we need to choose what ma? Cytokinesis, that is option C. I hope this is clear. Yes. Next question. Next question, that is assertion and reasoning. He asks you, like, the suitable stage to observe chromosome is metaphase. True, that is the metaphase stage where we can analyze the structure or morphology of the chromosome more clearly. In anaphase though, we can analyze the different shapes of the chromosome. Now if you see in reason, in metaphase, chromosomes are scattered in cytoplasm, which is wrong. The chromosomes are aligned in an equatorial plate, wherein asters, which are at the 2 opposite poles, will radiate the spindle fibers. Those are attached to the kinetochores, which are present at the centromeres. Okay, they are attached to what ma? Kinetochores, which are disc shaped structures over the centromeres, and with the spindle fibers they are attached to asters. This gives an alignment of a plate which is called as equatorial plate. What is it called as ma? Equatorial plate. Okay. So yes, reason is false and assertion is true. A is true, R is false, option C. What is correct option? Option C. Next question if you take, which of the following structures is found in a prokaryotic cell, he is asking. Mitochondria is not seen in prokaryotic cell. Golgi complex, no. Lysosomes are also not found. What is seen? Ribosomes, that are 70S ribosomes. 70S ribosomes are found in the cytoplasm and few ribosomes attached to the plasma membrane of a prokaryotic cell. And here in the place of mitochondria, mesosomes, which are membranous extensions, they are present in the prokaryotic cell, where they will release the enzymes which are which conduct the function of respiration. Okay, clear? Next question. Here again a table is given in the year 2022. So you can expect most of the match the following questions from this chapter. Okay. Here Leeuwenhoek, we know he identified the living cell. So here option is 4. Robert Brown, he gave us the nucleus, observed the nucleus in a cell, option 5. Next Schwann, he observed cell wall. Though he is a zoologist, he observed the unique characteristic feature in the plant cell, that is cell wall. Here it is option 2. Schleiden, he has observed different kinds of cells forming the tissues in the plant. Rudolf Virchow, who modified the cell theory, saying that all cells arise from the pre-existing cells which come under omnis cellula e cellulae. So here Rudolf Virchow, it is 3. Now you tell me what is the correct option. A4, B5, C2, D1 and E3. Option D is your correct answer. Next question. Assertion, if you see, growth in the primary wall of a young plant cell gradually diminishes as the cell matures, correct? Secondary wall is formed on the outer side of the cell is what explained in the reason. This is wrong. If you carefully observe a plant cell, the first and foremost layer which is formed between two plant cells at the time of cell division is middle lamellae. What is formed beta? Middle lamellae. Okay. The first and foremost layer formed is middle lamellae. And the next layer which is formed inside the middle lamellae is primary cell wall. What is that? Primary cell wall. Primary cell wall is extendable and this diminishes as the cell is maturing. The second formed layer is primary cell wall. And inner to the primary cell wall, closer to the plasma membrane, secondary cell wall is formed. How many layered is that secondary cell wall? Secondary cell wall is around 2 to 3 layered. How many layered beta? 2 to 3 layered. Okay. Third is what? Secondary cell wall. Okay. So now if you see the proper organization of different layers of the cell wall, first it is what? Plasma membrane. Next to the plasma membrane, what is there? Secondary cell wall. Next to the secondary cell wall you have primary cell wall, and at last on the outer side you will find middle lamellae. What do you find? Middle lamellae. Clear? So this is what you need to remember. Very important. Asked multiple times. This concept is asked multiple times in different ways. Very important beta. Keep it in your mind. Next question. Here if you see assertion, cytoskeleton is eukaryotic cell is made up of network of filaments, lipids and proteins. He is saying no, it is a network of proteinaceous filaments. What is it? Proteinaceous filament. Proteinaceous filaments. Okay. Next, reason. Cytoskeleton is composed of microfilaments, microtubules and intermediary filaments. Correct. So reason is true and assertion is false. In that case, the option will be D. Next question. Tonoplast, which is the outer layer or it is the membrane of the vacuole, right? This helps in osmoregulation of a cell. So here it is 4. Next, stroma, which is the fluid present in the organelle chloroplast. It contains enzyme related to dark reaction, and we can also say those enzymes are for anabolism as it is involved in the synthesis or construction of food material. Synthesis of food. Okay. So yes, here both the options are applicable beta. Both the options are correct only. Okay. Next, cistern of Golgi apparatus helps in modification of proteins and formation of what? Glycolipids and glycoproteins. And what, Nana? Glycoproteins. Okay. Now if you see, C is 1. Next, Plasmodesmata are the connection between two adjacent cells. If this is plasmodesmata, they are directly connecting the cytoplasm of two different cells, traversing the middle lamellae, primary cell wall and the secondary cell wall. Plasmodesmata are what? Cytoplasmic bridges that connects the cytoplasm of two cells, traversing middle lamellae, primary cell wall and secondary cell wall. Now, plasmodesmata is what? 3. It belongs to the option 3. Now, if you see 4, 2 or 5, anything is fine? Yes, 5, C 1 and D 3. Option A is your correct answer. Next question. Yes, if you see here, lysosomes are capable of digesting proteins, lipids and nucleic acids. True, lysosomes are digesting proteins, lipids and nucleic acids because they are rich in hydrolytic enzymes. They are rich in what, Nana? Hydrolytic enzymes. What are these hydrolytic enzymes? Proteases which digest proteins, lipases which digest lipids, nucleases that digest nucleic acids. Okay, so here reason is explaining assertion, then it is option 1. A is true, R is true, R is the correct explanation of A. Next question. Here if you see again a table in the year 2021. Keep noting the type of questions which are coming with her. Here circular DNA we need to correlate again the terms and the statements we need to correlate. Circular DNA observed in the prokaryotic cell, and in prokaryotic cell there is no membrane bound. So here you can keep the option 4. Along with option 4 here, plasmid DNA, that is extra chromosomal DNA which gives certain phenotypic character like resistance to antibiotics, that is given by plasmid DNA. No, this is also present in a prokaryotic cell, that is 3. Okay, so here what is there? 4 and 3. Next, fimbriae. These are bristle like fibers sprouting out of a prokaryotic cell. These are what, ma? Bristle like fibers sprouting out of the prokaryotic cell. Option 1. Next, this help in attachment of the bacteria to a substratum or a host cell, that is option 1. Next, middle lamellae is the outermost layer of the cell wall. What is the innermost layer? Secondary cell wall is the innermost layer. Which layer is present in between middle lamellae and secondary cell wall? Primary cell wall. Okay. So, middle lamellae, outermost layer of the cell wall, and it helps in, it develops from the cell plate. It develops from what, beta? Cell plate, which is initially formed during the cytokinesis of a plant cell. This is formed as a result of what? Golgi body secretion. As a result of Golgi body secretion. So, this is what? 4 again. Okay. Next, mesocarp and endocarp are fused in which type of fruits? Berry. Okay. Mesocarp and endocarp are fused in the berry type of fruits, and this type of fruit is observed in Solanaceae family member. Makoi is one of the example of this family member. And here what is the correct option? 3 and 2. Okay. 3 and 2. Now, if you see A, 4 and 3 correct. Next, 1 and 1, 2, 4, next 3, 2. Option B is your correct answer. Next question. Yes, nucleosome model, which is very, very important. Okay. So, in this question if you see, H1 histone molecule slides outside the nucleosome and seals the 2 turns of DNA. True. H1 or linker DNA is one and the same. H1 or linker DNA is one and the same. This is outside the nucleosome bead. Okay. So, this statement is correct only. Negatively charged DNA is negatively charged because it has phosphate molecules, phosphoric acid, right? Phosphate molecules. So, it is what? Negatively charged. And Positively charged histone molecules. Histone molecules are positively charged because they have the basic amino acids. What are the basic amino acids? Arginine and lysine. Okay, they allow packaging of DNA. This is also correct. Each nucleosome has 6 histone molecules. No, each nucleosome will have 8 histone molecules. How many histone molecules? 8 histone molecules. 8 histone molecules. What are they? H2A, H2B, H3 and H4. 2 sets of these 4 histone proteins are present in the nucleosome and they have 145 nucleotide base pairs. How many they have? 145 nucleotide base pairs. True. The DNA between 2 successive nucleosomes is called as linker DNA, which is also called as H1 linker DNA. True. Okay. So, if you see which is the wrong statement, C. What are the correct statements we need to take? A, B and D. Option 4 is your correct answer. Next question. Here if you see assertion, in a vacuole the concentration of ions, again in 2021 it is asked C. In a vacuole the concentration of ions, other materials will be less than the cytoplasm. No, the concentration of ions and molecules in a vacuole will be high concentration. Okay, and pH will be low, that is acidic in a vacuolar lumen. Okay, in what beta? Vacuolar lumen. Okay, next, tonoplast facilitate the transport. So, this statement is wrong. Tonoplast facilitate the transport of ions and other materials against the concentration. Right now in cytoplasm the concentration will be low and in vacuole the concentration will be high. So, there will be the movement is against the concentration gradient, which is called as active transport. Active transport which requires ATP. Which requires what? ATP. Clear? So, this is true. Reason is true, assertion is false, then it will be option D. Next question. Yes, here if you see, we need to fill the proper statements in the blanks given in this paragraph. Okay. So, yes, look into that properly. Each chromosome have a dash or dash. Each chromosome will have what ma? Centromere or primary constriction. Each chromosome will have what? Centromere or primary constriction. Centromere or primary constriction. On the sides of this we have disc shaped structures. What do we have? Disc shaped structures. What are they called as? Kinetocores. Kinetocores. Okay. Next, so here what option will come? Here it is centromere, C or D, and disc shaped structures called as kinetocores, that is G over here. Next, the dash chromosome has 2 equal arms. 2 equal arms are present in metacentric chromosome, means option A. Dash chromosome has 1 shorter arm and 1 longer arm in submetacentric, that is E. Okay, that is what beta? E. Dash chromosome has no second arm, that is telomere, where one is very negligible and one is long arm, that is telocentric B. Okay, the secondary constriction develops a small fragment. The secondary constriction will usually develop, this is the secondary constriction, which develops a small fragment, that is called as what? Satellite DNA. Satellite DNA, which consists of most of the repetitive DNA. Repetitive DNA fragments which are used in DNA fingerprinting, that we will learn in mol bio, no problem. F. So, if we look into the options, here DC or CD anything is fine, then G, then A, then E, then B, lastly it is F. Option B is your correct answer. Next question. Here the question is about common characteristics between both prokaryotes and eukaryotes. In prokaryotes, if you take, cytoplasm is there, and in eukaryotic cell also cytoplasm is there, wherein cytoplasm is the main arena of all the cellular activities that keeps the cell in a living state. Next, plasma membrane you will find both in prokaryotic cell and eukaryotic cell. Mitochondria you will only see in eukaryotes, which is equivalent to which organelle of prokaryotes? You have to tell me, students. That is what, Nana? Mesosomes. Mesosomes of prokaryotes. These are what? Membranous extensions of plasma membrane, which will secrete the enzymes that are respiratory in function. Next, cellulosic cell wall you will find only in the plant cell. You will find in the plant cell. What type of cell wall you will see in a prokaryotic cell? Peptidoglycan. Peptidoglycan or murin composition of cell wall in a prokaryotic cell. Meiotic cell division you will see in a eukaryotic cell. Amitotic cell division or amitosis, which is nothing but fission. This type of division you will see in a prokaryotic cell. Okay, so yes, only A and B are seen both in prokaryotic and eukaryotic. Your option will be B. Clear? Next question. Here if you see, choose the correct statements he asked. Okay, correct statements. Polar molecules cannot pass through non-polar lipid bilayer. If you take the lipid component which is in the plasma membrane, you have a, what do you have, beta? You have a polar hydrophilic head, polar hydrophilic head, and tail which is non-polar and hydrophobic, polar hydrophilic head and non-polar hydrophobic tail. Yes, so to transport polar molecules, definitely we need either carrier proteins or channel proteins. Yes, require carrier proteins. So statement A is true only. The plant cell vacuole plays important role in osmoregulation. Correct. Golgi apparatus are the site of synthesis of proteins and glycolipids. Wrong. They are modifying the proteins and lipids, and they are synthesizing glycolipids and glycoproteins. They are synthesizing what? Glycolipids and glycoproteins. Next, plasmid DNA shows resistance to antibiotics. True. Correct. On cell maturation, the secondary wall is formed as deposition towards the outside. No, it is formed towards the inner side of the primary wall. No, this is also wrong. So what are the correct statements? A, B and D. Okay, A, B, D. That is option B. Next question, ma. Next question, which is very simple, asked in 2020. Which of the following synthesizes the cell wall? Which of the following synthesizes the cell wall? Golgi bodies, right? Golgi bodies will synthesize cell wall, where initially it is synthesizing cell plate during the cytokinesis of a plant cell, which later this cell plate turns to what? Middle lamellae. From this, the subsequent layers which are formed towards the inner side of the plant cell are what? Primary cell wall and secondary cell wall. So initially, which organelle is helpful in the synthesis of cell wall? Golgi apparatus. Next question. Here again a paragraph is given, and we need to check out the relevant statements for it, relevant terms for it. Okay, fill in the blanks, asked again in 2020. Okay, Schleiden. Who is Schleiden? Schleiden is a botanist, German botanist. Okay, so 1 is F. Next, observed that different plants are composed of cells. Plants are composed of different cells. Schwann, that is 2. He is a British zoologist. He is a British zoologist. Together they propose cell theory. So this is option 2 is B. He observed animal cells and reported that plant cells have what? Cell wall. 3 is option D. And all cells have cell membrane or plasma membrane. He said all cells will have plasma membrane, and specifically unique feature of plant cell is having cell wall. Next, it is Virchow who modified the cell theory, saying that cells divide and form new cells from pre-existing or existing cells one and the same. He said pre-existing cells, but existing cells meaning also same only. This constitutes the cell theory, that is option A. Now you check out the proper flow of the options. F, B, D, E, C and A. Option D is your correct answer. Next question. If you look here, again it is the nucleosome model. See, it is very important, I told you, no? Again in 2020 also this question is repeated. Arrange the following in the ascending order of numerical number, he said, very important. Please do observe it keenly. The number of peripheral fibrils in a centrosome, he asked. What is a centrosome? Centrosome is again non-membrane bound organelle. You will see it in the animal cells only. Each centrosome will have 2 cylindrical structures called as centrioles. Each centrosome will have how many centrioles, beta? 2 centrioles. Okay. Each centriole will have what type of arrangement of microtubules? 9 plus 0 arrangement of microtubules. What is this 9 plus 0 arrangement of microtubules? Wherein at the center you will have proteinaceous hub. To this proteinaceous hub, 9 radial spokes will be formed. Okay, 9 radial spokes. Each radial spoke will have 3 microtubules. Each radial spoke will have how many microtubules? 3 microtubules like this. Okay, like that if you fill on this circle, how many peripheral microtubules you will get, beta? How many peripheral microtubules? Yes. How many peripheral microtubules? 9 radial spokes, right? 9 into 3 are 27 peripheral microtubules you will get. Okay. The total number of microtubules in axoneme of cilia. What are cilia? These are ores that emit from the cell surface, right? Ciliary bodies. For example, paramecium. If you take the core of this and see, again you will find a microtubular arrangement. This is what type of arrangement? 9 plus 2 type of arrangement, where you will observe like this, wherein 2 central microtubules will be there. 2 central microtubules will be there. From this, which are covered in the central sheath, 9 radial spokes are emitted out. 9 into 2. Okay. For each radial spoke, how many peripheral microtubules are there? 2. So, on total you tell me how many microtubules? 2 plus 18, 20 microtubules you will observe in the axoneme of cilia. Clear? Now, number of arms in telocentric chromosome he is asking. If you take a telocentric chromosome wherein one arm is minute or negligible and the other arm of the chromatid only, you will see. So, what are the number of arms you will find out in telocentric chromosome? That is 1 only. The number of nucleotides in a nucleosome he is asking. So, if you take a nucleosome bead, or a complete nucleosome, you will find out, like in a typical nucleosome, approximately 200 base pairs are present. How many base pairs, beta? 200 base pairs. What is a nucleotide? Nucleotide means it is a composition of pentose sugar, which is attached to a nitrogen base and then a phosphate group. This combines a nucleotide, right? So, how many bases are there? Number of bases are equal to number of nucleotides, correct now? So, number of base pairs are there. How many base pairs? How many bases from this base pairs? 400 bases. Okay. If there are 200 base pairs, from this how many bases we have? 400 bases. number of nitrogen bases are equal to number of nucleotides, that is 400 nucleotides. So, the correct order which is not given here, you see the correct order is C, that is 1 telocentric chromosome, then it is axoneme of cilia, that is 20 microtubules, okay, that is B. Next comes the peripheral fibrils of a centrosome, that is A, and then last one, that is D, okay. C, B, A, D is the right option, okay. It is not given over here, but this is the right option. Next question beta. Next question, a typical nucleosome bead he is asking. A typical nucleosome bead means only this. What is the typical nucleosome bead? Only this region. If you see how many is made up of how many histone molecules and base pairs he is asking. Of course, we have already discussed it is of 8 histone molecules and 146 base pairs of DNA. Option A is your correct answer. Next question, it is again with respect to match the following. Here if you see amyloplast will store what, Nana? Carbohydrates or starch, that is 3. Okay, elioplast will store oil and fats, that is 4. Aleuroplast will store proteins, that is 2. Leucoplast will store just leucoplasts are what? Under leucoplasts only all these 3 comes, no? Amyloplast, elioplast and aleuroplast. Leucoplasts are colorless plastids of varied shape, size and stored nutrients, that is 1. Now if you see A3, B4, C2 and D1, option A is your correct answer. Next question. Here again statements are given in the year 2019, okay. If you look into it properly, just a minute, which says the following characters are found in ribosomes. We need to choose the correct characters of ribosomes. They are composed of RNA and proteins not surrounded by any membrane. Absolutely correct, okay. Ribosome is composed of RNA and proteins. The membrane transports a number of ions, wrong. They are membrane bound vesicular structures and rich in hydrolytic enzymes, wrong. Ribosomes are not membrane bound. They help in synthesis of proteins, true. They help in what beta? Synthesis of proteins. So, the correct option is A, 1 and 4, okay. They do not have membrane only, option 2 is wrong, okay. So, the correct option is 1 and 4, option A. Next question. Here if you see again in the year 2018, mesophyll cells are given, different types of cells are given and their shape is given in the other column. Mesophyll cells are round and oval, option 3. Tracheids cells elongated, option 4. Nerve cells are long and branched, option 1. White blood cells are amoeboid, option 2. Okay. Now if you see 3, 4, 1 and 2, option C is your correct answer. Next question. Next question. Last and final question of the year 2018. If you see, just a minute. Yes. If you see, the fluid mosaic model proposed by Singer and Nicholson indicate the quasi-fluid nature of lipids and enables lateral movement of proteins in the overall lipid bilayer. True. Assertion is true. The quasi-fluid nature of lipids enable the lateral movement of proteins. Correct. In the reason, the cell wall of the young plant cell, the primary wall is capable of growth which gradually diminishes as the cell matures, correct, and the inner cell wall is formed on the inner side of the plasma membrane. Reason is also true, but reason is not explaining A. In that case, what will be our option? B. Both assertion and reasoning are true and R is not the correct explanation of A. Option B is correct. Yes. With this, we have completed all the questions from this chapter from 2024 to 2018. This is a very important session. All the aspirants who are aspiring for APEAPCET must watch. It is useful for you people. Okay. In the next session, I am coming up with the NEET PYQs of this chapter. Thank you.